To explore how the fabric of this universe may explain not only material reality, but also the experience of that reality by consciousness, this is Neon Galactic. I'm James Falk with co-host, educator, and fellow traveler David Stuss. Thanks for joining us. In many ways, it's a given that human consciousness was the first frontier of organized human investigation, a pursuit that birthed a long line of ancient esoteric wisdom communities dedicated to the study of mind, the self, and their relationships to the broader cosmos. But things change. As time went on and the sciences shied away from subjectivity to put more and more focus on the material world as arbiter of truth, such research lost its luster and became supposedly anathema to the scientific method. So here we are now, all these centuries later, and still the questions of consciousness vex us, resisting explanation and making up what's become known in the field as the hard problem. Is there any more fundamental human question than how the odds and ends of material existence, the human body, and wider physical world might combine to eventually produce the exquisite complexities of sensory experience? Solving this mystery would go a long way toward answering so many of the fundamental questions about human existence, spiritual possibilities, anomalous phenomenon, and the potential meaning, if any, to life. Might it be possible that consciousness is an intrinsic part of the universe, not just an epiphenomenon, and that evolution itself might have been drawn forward by a cosmos in search of positive experience? These are big questions, so where do we look for answers? Since the discovery and mapping of the quantum world, we've seen many of our seemingly long-settled assumptions about reality toppled in favor of a much more holistic, connected, and mysterious set of fundamental truths. The quantum underlies every aspect of the physical world, and its nature remains a subject of much debate. Could the two questions of human consciousness and the quantum connect? Could the seemingly wet and warm environments of the human brain host quantum processes? And might those processes help explain the wickedly complex workings of the human mind? Or is that very question just a sleight of hand to solve one mystery by drowning it in another? Our guest today is an innovative thinker, a maverick in the field of consciousness, and someone who has confronted the mainstream scientific establishment with a theory that seems to be gaining more and more credence as time goes on and evidence mounts. Retired University of Arizona professor and anesthesiologist Stuart Hameroff, along with physicist Roger Penrose, developed the Orchestrated Objective Reduction theory of consciousness. He joins us today to discuss the idea, the proof, the battles he's fought to advance the theory, and above all, the ongoing mystery of consciousness and what that might reveal about the cosmos. The human brain, neurons, and consciousness itself may be infinitely more capable and astounding than we've ever expected. And if so, Hameroff may have the why of it. Welcome, sir.
Thank you, James. Nice to be here. Thanks for having me.
Yeah, no problem. This is an absolute privilege. And we're joined by David Stuss, my good friend and occasional co-host. Anything you wanted to add before we get into the questions, David?
Oh, just delighted to be here. I've been tracking Stuart's work for many, many years, starting back in the 90s. And yeah, this is a real privilege for me too. Thank you.
Yeah. So, Stuart, if you don't mind, we'll start off with microtubules. It's something I didn't mention in the introduction, but that's a key part of your theory of consciousness. Can you explain for folks who may not be swimming in this kind of material all the time what a microtubule is, how it came to be a specialty of yours, and then how it fits into the question of consciousness?
Right. Well, all living cells have an internal skeleton, kind of like our bodies have bones. And the cytoskeleton in cells is comprised of structures, including microtubules, which are cylindrical protein polymers that self-assemble from a protein called tubulin, a peanut-shaped protein that has a positive and negative end. And these things actually self-assemble into fairly long, in some cases, cylinders, which have crystal-like lattice structures, and they make the main part of the skeleton of the cell, but they're also the nervous system of the cell. So imagine if your bones also were your nervous system combined, and that's kind of what happens inside cells. There are other parts of the cytoskeleton. There's actin protein, centrioles, which are kind of the mothership of the cytoskeleton next to the nucleus and kind of where all the microtubules radiate from. And microtubule associated proteins that can connect one microtubule to another, that can walk along the microtubule carrying synaptic cargo for synaptic plasticity, uh, and others like tau, which is prominent when you get Alzheimer's disease, when your microtubules fall apart, that may stabilize the microtubules, but they also may be traffic signals to tell the motor proteins where to get off and deliver their cargo. So their placement is kind of like memory. And I think microtubules actually encode memory because most people in neuroscience would say, well, memory is in the synapses, and it may be to some extent in the synaptic protein. So the more the more a synapse passes information, it's going to solidify that pathway through a network. And most people think of network effects as necessary, but the synaptic proteins only last hours to days, and memories can last lifetimes. So it's much more likely that the memories in dendritic microtubules, which have a huge capacity. But we can come back to that later. The microtubules organize stuff inside the cell. So if a neuron grows, like it's a microtubule polymerizing and pushing the actin ahead of it, and to make a new synapse or to branch or to make a connection here or there, wherever it goes. And when cells divide, now neurons don't divide, which is actually a very strategic point, which I'll come back to. But when most cells divide, the microtubules, whatever they're doing, will depolymerize and reassemble in mitotic spindles, which are necessary for mitosis to pull the chromosomes apart in an exact duplicate of each other. So you have the same chromosomes in the two daughter cells. And after they form their own cells, then those microtubules go back to being whatever they were doing in a kidney cell or a liver cell or whatever. Neurons don't divide, so and in dendrites they're capped, so they have the same lattice arrangement over your lifetime, basically. So and there's about a billion, maybe 100 million to a billion tubulins per neuron, and each one can be in about 30 states, genetic or post-translational. So the information storage capacity in the microtubules is what, 30 to the billionth, or a billion to the 30th, I forget which, but either way, it's a huge number per neuron. So another way to look at that is that if you think the microtubules are oscillating in at megahertz, let's say, which we know that they are, or 10 megahertz, then and there's a billion of them, that's 10 to the 16th operations per second per neuron. Now, the singularity, remember Ray Kurzweil and talking about when we reach brain equivalence, we'll have a singularity. Well, the brain equivalence they calculated was based on about a billion, sorry, 100 billion neurons with 1,000 synapses at 100 hertz, which gives you their calculations, 10 to the 16th operations per second for the whole brain. And so in the 80s, I was going around to neural net and AI meetings saying, no, 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 the information capacity is actually 10 to the 16th operations per neuron. So your target, your goalpost is way, way, way downstream.
Yeah.
Well, they didn't like that very much. And they basically told me to get lost and to F off and a few other things. Because, they're trying to, get a few more billion out of the government to make conscious computers, which of course didn't happen and hasn't happened, probably won't happen, at least in Silicon. But so I was, I had gotten interested in the microtubules in medical school, and then went into anesthesia and started studying how anesthesia works. Turns out they act on microtubules, so that's another story that we can come back to. But I did some modeling of microtubule information processing with physicists in the late 70s and 80s, and like I said, was going around these meetings being a pain in the butt to the AI guys. And then one day somebody said, okay, Mr. Wise ass, let's say you're right, and that explains consciousness. Or sorry, that gives you all this information and processing. How does that explain consciousness? How does that explain, pink and love and envy and qualia and all that? Basically, the hard problem, which hadn't come out yet, at least Dave Chalmers' version. And I took the point very seriously. I said, I don't know. That's a very good question. So, uh, fortunately, that person recommended I read a book by Roger Penrose called The Emperor's New Mind, which had a mechanism for consciousness. And so I did, and I read it, and I was blown away by it. And he in the first part, he talked about consciousness had to be something non-computable because of Gödel's theorem that a mathematical theorem can't prove itself, that you need something outside the system, like a mathematician. And in the case of the brain, if everything's a classical computer, you need something outside of classical physics to be involved in judging or understanding or gauging whether something is true or not. Understanding is a good broader term for that. And for that, he said you had to go outside physics, outside classical physics to quantum physics and collapse the wave function because we don't understand what guides the collapse of the wave function. And some people question whether there is a collapse of the wave function. So that got into the measurement problem in quantum mechanics, which I hadn't thought about too much back then. But when I read the book, I thought, I didn't really understand all this, but he's onto something, and he's obviously very smart, and he's got a mechanism, and it's the only mechanism I'd ever seen, and still to this day, 30 years after I read the book, I haven't heard another specific mechanism for consciousness other than emergence, hand wave, emergence, complexity, hand wave, blah, blah, blah. Or integrated information of pure numbers that don't have any biology. So as far as I can tell, it's the only it was and remains the only specific scientific mechanism. And of course, but he didn't have a biological device in the brain that could do such a thing. And he knew that things in quantum technology needed to be done at absolute zero temperature to avoid decoherence. So he knew there was going to be some issues, but he said there must be something that can do this in the brain. But he said, I have no idea what it is. If anybody out there has an idea, let me know. So I was reading this book and I go, yeah, I got an idea. It's microtubules. I've been studying microtubules 20 years at that point, and I wrote him an old-fashioned letter, and he wrote back and said, yeah, uh, let's talk because I was going to England for something else, and she invited me to Oxford, and so I went, and he met me at the train station, went to his office, and he said, "So tell me about these microtubules. They're not simulations, are they? They're real." I said, "Oh yeah, they're definitely biologically real." And I handed him a book that I had written about microtubules in 86, 87, and this was about 92, I think, that when we met, and a bunch of papers, and we went through all the pictures, and he was particularly taken by the Fibonacci geometry of the microtubule A lattice. And so we went over that, and he mentioned he was going to a conference at Cambridge with philosophers Dan Dennett and Pat Churchland, both reductionists, materialists, eliminativists, and they already didn't like his ideas and had let it be known. And anyway, I said, "Oh, I'd like to go to that, but I got to go to my meeting, what I was there for." And so he said, "Well, thank you," and we shook hands and said goodbye. And I walked away. I said, "Well, that was cool. I got to meet Roger Penrose," thinking, "Well, that was the end of that." But when I got back to London a couple of weeks later, on my way home, my friend had been to the conference in Cambridge. He said, "Well, Roger Penrose was talking about you and your microtubule idea." And I said, "Wow, that was pretty cool." And soon after, I got invited to a meeting in Sweden that he was involved with, and we met up above the polar circle and midnight sun. It was just a gathering of scientists, no audience or anything, and started collaborating and figuring out how we could apply his idea, which was called objective reduction, a collapse of the wave function due to quantum gravity, uh, to microtubules. And I said, "Yeah, we got to do that." I had no idea how, and we started talking about it, and it took a while, and eventually in '96 our full theory came out. We had another paper before that because Pat Churchland wrote an attack piece against us, a very smarmy, obnoxious, sarcastic, uh, which is her style if you know her. And we had to respond to that. But and then the full theory came out in '96. And it was immediately criticized. The brain's too warm, wet, and noisy. Everybody knows that. You guys are full of crap and forget about it. There's no collapse anyway. And what are you even talking about? And blah, blah, blah. So, it went over like a lead balloon initially, although people said, wow, that's different and that's interesting. And, we had to face the warm, wet, and noisy problem, which we finally did figure out. And now there's pretty good evidence. There's very good evidence for it. And it turns out anesthesia acts by quantum effects on microtubules. There's good evidence for that. So all in all, and some other stuff I could talk about, all in all, I think there's more evidence. We haven't proven collapse yet. Collapse is a tough nut to crack. But the conditions for collapse that we need, which is quantum coherent superposition at warm temperature for a period of time, T, to be determined, and I'll come back to that point, I think it's all been proven pretty well. So I claim that, and I say this in interactions with other scientists, philosophers, people I debate with, discuss with on X, Twitter, and at conferences and wherever. I claim that our theory, which is called Orchestrated Objective Reduction, ORCH-OR, has more supportive evidence, has more explanatory power, biological connection, and experimental evidence than all the other theories, which I call cartoon neuron theories, combined. And those, because those theories, like global workspace, higher order thought, to some extent IIT, although IIT doesn't even have neurons, it's just pure numbers, which is I think silly, but they call it a fundamental theory. I don't buy that. And then predictive coding and recurrent processing, which I like because predictive coding and recurrent processing can happen at multiple scales. And I think the key is the problem with cartoon neurons are at one scale, hertz, EEG membrane activity, up to 100 hertz, maybe 40 hertz, maybe 100, 150. But actually, it turns out microtubules have oscillations in kilohertz, megahertz, gigahertz, and terahertz, every three orders of magnitude, like a fractal. In fact, we're calling them time crystals. So microtubules seem to be particularly built for this, or I don't know if it's a question whether they evolved to do that or it's just part of nature. But in any case, they can handle all these frequencies both sensing and actuating, which is perfect for biology because, we have very tiny stuff going on in our bodies. We have fairly large stuff. And like a hologram, you want the same information at every level. And I think that's what's happening. So that's kind of an aerial view of how we got here and where we are now. And like you said, there is more and more evidence. So we figured out how to do experiments looking for quantum effects. And we've shown warm temperature quantum effects in a microtubule that's inhibited by anesthesia, which has got about 1 million times more evidence than any other theory because they're all, the other theories, they like in the big showdown between global neuronal workspace and IIT is part of the Templeton program, which I'd be happy to talk about because we were in that too. Um, what the whole prediction was in a $5 million experiment because they had hundreds of patients and MRIs and EEGs and blah 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 MEGs, $5 million. And one theory predicted they'd see MRI activity posterior, and the other theory anterior. Which and in the end it was inconclusive. Well, sometimes they see it here, sometimes. So it was all a big waste of time. And even if one had come out, if you have MRI activity in one particular place versus another, what does it tell you about consciousness? Nothing, because MRI is apparently metabolic related in blood flow. And so ironically, what's metabolically active and consumes energy in the brain are membrane depolarizations and cognition. And consciousness is actually a very low energy process because it's a quantum process in the microtubules. And one example of that was given by Robin Carhart Harris, who's a famous psychedelic researcher. He's now at UCSF. But back in 2012, he was still in London, and he gave a talk at our conference on psychedelics. And he had this experiment. This is a really great experiment. He had patients sitting in an MRI scanner or an EEG or an MEG actually, and with an IV, and they got intravenous psilocin, the active ingredient that you give IV. And they were told to lie there peacefully with their eyes closed, don't look around, keep your eyes closed, don't talk. Tell us later how you were feeling, what you were experiencing while you're getting the psilocybin. We're going to scan your brain and check your MEG, EEG. And they kind of expected to see the MRI and MRI in particular light up like a pinball machine because of all this tripping activity, and the MEG also to be quite active. And so they did the experiment, and subjects reported later that they were vividly hallucinating and having this amazing psychedelic experience, but yet their MRI was cold and dark, like they were comatose, and their EEG, MEG was almost flat, like they were, you know, brain dead or comatose or something. And yet they were vividly hallucinating. So I remember I chaired the session for Robin, and he had this explanation by or entropy, which he's still saying, which I actually don't agree with, but it could be right. But what I said was that what really burns the energy of the membranes, cognition. If you're just laying there, your membranes are on vacation; they're not doing anything. But you're conscious as hell. You're highly conscious because the microtubule quantum stuff is going crazy at a deeper level, which you found out later is probably megahertz and gigahertz, um, more quantum activities, and that's where consciousness was. But you had no cognition. You didn't have to interact with the outside world, so your membranes could rest, and you didn't need ATP or anything. So the MRI was cold and dark. So I don't think the point is that the experiments like the Templeton stuff, GNW versus IIT, and the rest are pointless because just knowing where an MRI lights up doesn't really tell you, A, that consciousness is actually happening there. And even if it is, what is causing the consciousness? Is it the blood flow? I mean, that'd be kind of a hard sell. Um, Probably the best explanation for why they're and the bold signal from MRI, blood oxygen level dependence, comes after the activity. So it's not, signaling the activity. It's happening after the activity. And nobody's quite sure why. Well, you need more nutrients or blood supply, but that didn't really add up. And one explanation I heard was that actually it's to cool the brain. It's to cool down that little region of the brain after something's happened. And that would be especially important in a quantum mechanism. But the point is that the other theories have zero evidence, uh, and we have a fair amount, and yet we're still the oddball, we're still the underdog, which is fine. I don't mind that. But I think we're on the right track. And the anesthesia story has come around towards microtubules. That's kind of a whole nother story.
Let me ask a couple of questions just to get a sense of how the brain works. You mentioned that there's cognition, but then there's also consciousness, and they're two separate things. It sounds like you're thinking that microtubules are responsible for generating the conscious experience of the mind, of the brain, but then cognition is a neuronal thing. Is that fair to say?
Well, I
Would you
think
agree with that?
microtubules may be involved in cognition too, but they need to be classical. Sorry, they need to be quantum. They can do it with classical. They can do classical information, and I think it could be that cognition, at least not well, non-conscious cognition, could be strictly membranes. You're driving to work. You go the same way every time. You're daydreaming, or at least I am, about what you're going to be doing when you get there, or why your team lost, or whatever. And then all of a sudden somebody swerves in front of you and become conscious. You again become conscious of the road and you're driving. So I think you're we can be on autopilot where the membranes are active and the cognition's going on, but you're not necessarily conscious of it. But it can quickly draw you back in if need be. So yeah, I think there's a distinction there. The other way to express that is that in anesthesia we use evoked potential monitoring, for example, for spine surgery. So if the surgeon's operating on the C spine from the front or the back, and you want to make sure that he or she isn't, literally screwing the spinal cord with a screw because they're putting screws in the bones and or causing anything to impair spinal cord function, we have these electrophysiology people who come in and put electrodes on the feet and the hands and the scalp, and they can stimulate the feet or the hands and measure the signals from the scalp from the brain to know that there's signals passing through the spinal cord, or and they can stimulate the brain and make the foot twitch or the hand to know that the motor is going through, and yet the patient's unconscious, the patient's under anesthesia. So the point is that the brain's still active under anesthesia. There's EEG, it's just slowed down. You have evoked potentials, which we use for monitoring. And neurons fire if you are able to stimulate a neuron, it will fire. Uh, but there's no consciousness. So what's happening at the membrane, which is what we can easily measure, may or may not be related to what's going on in consciousness. It could be, but it doesn't have to be. And there are cognitive things without consciousness. There's conscious things without cognition, like in the psychedelic in the psilocybin experiments I mentioned. So yeah, they're not the same. And then it's not just a gradation when you get really cognitively excited, you become conscious. No, you can be conscious at a very low, subtle, almost, well, it's perceptible level. So I think they're different, and I think that's a hard lesson. It's kind of like behaviorists gave up on consciousness to measure what they can measure, which is cognition and behavior, what happens when you move stuff or you respond. So they weren't measuring consciousness, and they knew that, but they didn't care. So they made consciousness a dirty word and a dirty concept, and just focused on what they could measure to bring psychology back into science in their view. I think that was their motivation.
Is there evidence that you can point to where the activity of microtubules is interrupted by anesthesia? Is that something that's been able to be measured?
Yeah, yeah. So anesthesia is a very interesting story, and I think probably the key to this whole thing because anesthesia is selective. As I just said, it takes away consciousness but spares neural activity, including evoked potentials and cognition, which can continue. So where and what does it actually do? So in the 1800s, 19th century, excuse me, these gases were discovered which if you inhaled a small amount of them, small amount, people got euphoric, giddy. And this was like ether, diethyl ether. So they had ether frolics, parties based on sniffing ether, and then laughing gas, nitrous oxide. And if you get all giddy and goofy, and that's still used, particularly in the dentist. We don't use it too much in the OR very much, but it's used in dentist office and stuff. And I used it a lot when I was in anesthesia, or at least early in my career. Anyway, so if anesthesia is selective for consciousness, what does it act on, right? Where? That's the key. That's the best way to figure it out. Figure out. Well, anesthetic gases are inert, pretty much. They don't form chemical bonds. All the drugs we give, the other drugs we give that are polar. They have charge, and we give them IV, or that you take by pill and it gets absorbed, or you give it IM or some other way, and they're polar, soluble in water, soluble in fluids because they have charges. Um, and they go to charge, they find opposite charges. So they got a positive charge, they find a negative charge, like on a receptor, on the outside of a cell. They don't have to even go inside the cell, and that's how most drugs work. Anesthesia, the gases, they're well, how do you know where a drug goes when you give it? It's based on its solubility. So all the polar drugs are soluble in charged areas, in water, in blood, or anywhere there's charges. And it's chemistry. So the charges interact, and they do chemistry, and they bond in one way or the other. The anesthetic gases are inert, and they don't form chemical bonds at all. In fact, one anesthetic gas is xenon, which is an inert element, a gas. And but all of them have, and they're all chemically different. This is kind of bizarre. They're chemically different. They can be ethers, they can be inert elements like xenon, they can be halogenated hydrocarbons, and then there's some IV ones. But all the gases they share one feature, which is at least a surface or the whole atom in case of xenon that is inert and has a full electron outer shell, so no electrons to share with other atoms or elements or molecules. So what happens is if you have two completed electron outer shells, um, and like for example, two benzene rings, so the aromatic rings of benzene. I'm going to digress for a second, but um, life is based on organic chemistry. People say organic carbon chemistry. And people have been saying, like David Chalmers is trying to backtrack on the hard problem now, and saying that AI can be conscious and AI will inevitably be conscious because there's no fundamental difference between silicon and carbon. To that, I say bullshit, because first of all, it's not just carbon, it's organic carbon. And what that means is uh, aromatic rings. So, benzene, like what goes in our gas tank, it's six carbons. Let me back up. In the 18th century, chemists were working out all the hydrocarbons, which are linear chains of carbon and hydrogen, and they could have one or two bonds, and they had a formula CN plus N or CN plus 2N uh, the number of hydrogens. But then they had C6H6, which didn't fit because they Kekulé, this German chemist Kekulé, supposedly had a dream that the linear hydrocarbons were snakes, and one of the snakes swallowed its tail and became a loop, a ring, the Ouroboros in mythical terms. And he
Yeah.
woke in the morning and said, "Aha, benzene is a ring," and he was right. And it's a hexagonal ring, six carbons, which means there are three extra electrons. So those electrons form electron clouds above and below the ring. These electron clouds are essentially quantum objects because they have electrons distributed all over all over this volume. So it's a volume of a pure quantum object, and yet it's stable. And a lot of energy, you put them all together in bulk, light a match, and they explode. That's how our cars run. But if you put them in an array, a geometric array where they're just barely touching, like in graphene, or in fullerenes where they make carbon nanotubes or buckyballs, or inside a protein where the aromatic amino acids can just touch at the van der Waals forces. What happens is the electrons in this one repel the electrons in this one, and the electrons oscillate back and forth. They couple and oscillate, and they oscillate in terahertz, 10 to the 12th hertz, which is infrared or optical. So they're actually giving off photons just during their oscillations.
Yeah.
And this is what happens inside proteins. In fact, this is how proteins fold. The aromatic amino acids attract each other. They get rid of the water. They don't want the water. Oil and water don't mix. And so they form these regions inside proteins that are all aromatic rings, which have quantum optical effects like fluorescence and so forth. And they get rid of the water. So from an anesthesiologist standpoint, we give our anesthetic gas through a mask and or a tube, and it goes in the patient. And where does it go? It goes to the non-polar regions where there's no water. These aromatic rings inside proteins, and that's where they act. So we know that the anesthesia acts in regions of non-polar aromatic rings inside proteins, which have quantum optical effects and quantum effects, and that's where they act, and that means that they must be since they act by quantum interactions, they bind where the quantum stuff is happening. They must be working by inhibiting quantum effects, and we've shown that experimentally that anesthetics bind in microtubules, but also in our experiment in the Templeton Foundation yeah, it was a contest actually, where they try to pair two different theories. We had a meeting with IIT, and we couldn't come to an agreement because I couldn't figure out how in the world you could falsify IIT, because I don't really think it's a real theory, but it's a whole nother story. So we gave up, but Templeton said, okay, we'll give you money just to do your experiment. What we want to do is demonstrate a quantum effect in a microtubule at ambient temperature, and then show that it goes away with anesthesia. And our adversaries, Christoph Kolk and Giulio Tononi, said, "Yeah, great. We'll do it inside a live monkey, and we'll put the monkey to sleep, and then see if the quantum effect goes away." I said, "No, you don't understand. This has never been done before. Even to do it at ambient, we have to do it on an optical bench with all these lasers lined up perfectly. You can't have any vibration. To do it you can't do it inside a monkey, not the first time. Maybe someday, but first we got to do it on an optical bench." And so that's what we did. And we did it at Princeton in Greg Scholl's lab. And our postdoc, Cara, went there and did this experiment where he hit a microtubule with UV light, and it fluoresces, but before fluoresces, the exciton propagates down the microtubule for a length and a period of time, and then emits a lower energy photon, fluorescence. And we gave these labels which fluoresced at a slightly different wavelength, so that when it propagated, when the exciton propagated, if it got to this, it would come out at a wavelength we knew was the AMCA. AMCA was the label. We could tell how far it went. And so the first thing we found was that it propagated farther than you can explain by a classical mechanism. It had to be a quantum mechanism. It propagated farther in distance and also persisted longer in time. So it was a room temperature quantum effect, which we showed in a microtubule, which was the first thing that first prediction. The second prediction was it would go away with anesthesia. We tried two different anesthetics, isoflurane, a gas, and etomidate, a soluble polar one. Both of them inhibited about the same amount. So we called our shots. We showed the quantum effect of room temperature in a microtubule, and it was dampened significantly by different anesthetics. So our experiment worked. I think it's the only experiment in the whole Templeton program that has shown any support for any theory. All the rest have been inconclusive and a huge waste of money and time. $30 million they poured into that. Our experiment was $100,000. IIT and they got 5-10 million total, and they're still spending that money on propaganda and promoting theories that have already been disproven. If we had been disproven, they'd have thrown us the heck out of science. But they just keep pushing out the same BS because they have big names behind them, I think. So anyway, a little pet peeve there, but it is annoying that all this money is just going down the drain, wasted on these stupid experiments, which aren't going to show anything. Even if you show MRI, I call it pin the tail on the brain map. They're just saying, oh, you're going to see an MRI here, you're going to see an MRI here. If it's here, you win. If it's here, we win. Bullshit, because you don't know, even if you predict properly, it doesn't tell you what consciousness is. What's it doing there? Where it is not the problem. I think consciousness can be all over the place or any place. Any particular place actually can move around. And even if it's not, it's distributed memory over a lot of the brain because it's entangled. So I think the idea of it's here or it's there, depending on firing here and there, is garbage.
Can I,
Do you have a question you want to ask?
Yeah, I'm curious, Stuart. As far as the aromatic interactions, I'm wondering if you looked at DNA at all. I mean, of course, one of the main stabilizing forces is you got the
Right.
pi stacking, all the way throughout
Right.
the nucleobases. So why proteins and not nucleic acids?
Well, I think DNA is also a quantum device, a quantum computer. I have a section in one of my papers about a model for quantum computing and DNA, because through the pi stack, as you just said, the helix. If you go down the core of the helix, it's one uh, uh, purine or pyrimidine after another one. So all these nucleic acids stacked up in the pi stack, and that's a whole linear quantum volume right there. So yeah, there's probably some kind of quantum communication going on between the DNA and the microtubules. But as far as moment-to-moment consciousness, I think it's in the microtubules with maybe, like memory banks or genetic instructions coming from the DNA. But I think biology in general is a quantum mechanism. I think, people, particularly the neurocomputationalists that I've been ragging on, they don't even think about what life is. And I think life is as big a mystery as consciousness. And, they kind of blow that off by saying, well, it's functionalism, metabolism, adaptation, homeostasis, blah, 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 blah. I'm going, yeah, but that's not life. Those are the functions that support life, the conditions for life. But what is life? And, we don't have a good definition. I think the vitalists were probably closest, but they got run out of town because they were calling on either electromagnetics, which people already knew about, or some unknown force. And but then Schrodinger, in his book "What is Life," actually raised the idea of a quantum vitalism, which I think is probably the right idea. I think the brain needs quantum coherence and entanglement for things like binding, spatiotemporal binding, zero phase lag gamma synchrony, how you can have 40 hertz over here exactly synchronized with over here. You can't do it by uh, axonal pulses or firings. You can't do it by ephaptic transmission because they drop off with uh, 10 to the 3 or 10 to the 6, I forget. So they're not going to get far enough. And I think entanglement's the only way to explain perfectly zero phase like gamma synchrony, and probably synchrony of a lot higher frequencies. My friend Anirban Bandyopadhyay at the National Institute of Material Sciences in Japan, in Tsukuba, discovered coherent oscillations of microtubules in hertz, kilohertz, megahertz, gigahertz, and terahertz, every 3 orders of magnitude, which is why we think microtubules are time crystals. And so what he did was, this is actually a really cool experiment. If this is a microtubule, let's say, and you put 4 electrodes on it, and two to stimulate and two to record, or however you work it out. If you just put a voltage across, it's an insulator. There's no current. But if you apply a voltage or an alternating current, let's say, or alternating voltage, however you want to describe it, and sweep the frequencies from 0 on up, it's an insulator. But then you get to a particular frequency, uh, let's say kilohertz, there's a huge jump in conductance, ballistic conductance. It may be quantum if you get rid of the interface, but you see 3 peaks, and each peak has 3 peaks, so it's a triplet of triplets. And okay, then you get higher frequency and they go away, and then you get up to, so that was killed. Then you get up to megahertz, you see the same thing, 3 orders higher. Then you get up to gigahertz, you see the same thing. Terahertz, you see the same thing. And you can measure triplets from the scalp. You can measure megahertz triplets from the temporal area of the scalp. So it's part of the EEG. And we're actually working on experiments looking for effects of anesthesia on the megahertz triplets. And that'll be reported on at the Science of Consciousness conference in Tucson in April. Some experiments that Anirban's team has done in Japan. So more and more evidence that and megahertz cannot come from membranes. Membranes can't depolarize that fast. So and even kilohertz. The Neuralink that Elon Musk's thing works in kilohertz. I think 19 kilohertz. And we actually had the one of the first recipients of it come talk to our consciousness club at the University of Arizona. And he had this horrible accident, and it was a quad from here down. And they put this thing in, and now he can, he can talk and he can move his eyes, and just by turning his head and fixating on something, it's like a cursor, and then he can operate a computer. And while he was talking, he was also playing chess with somebody in the audience on a screen who was from the chess club, so must have at least known how to play pretty well. And he beat him while he was telling us about his experience. It was really quite impressive, actually. The point is that 19 kilohertz is too fast for nerves to fire, or membranes to depolarize. So I said, well, why using that? Well, it works the best. Well, what do you communicate? Well, we don't really know, but that's what works the best. So they're probably communicating directly to microtubules without knowing it. And so that's kilohertz, and then you get up to megahertz, and that's probably where the real action is as far as consciousness, about 10 megahertz. You can also have gigahertz and terahertz, and people are putting photons into the brain, and it actually gets through somehow through the skull, and it seems to be beneficial if you pulse it at 40 hertz. And we've been using ultrasound because megahertz. I want to stimulate microtubules, but I don't want to put electricity in the brain. But ultrasound is megahertz in mechanical vibrations, and we've been using ultrasound in anesthesia forever. And I was very familiar with it. So when Audubon discovered all this, I said, "Well, gee, I wonder if we could megahertz." And I saw this ultrasound machine. I go, "Holy cow! Why don't we try ultrasound?" So I went to the literature to see if anybody would try ultrasound into the brain, and it was approved for imaging the brain. It was still used to image in newborn brains through the fontanelle where there's no skull to look for bleeds. And well, how damaging can it be if they're giving it to newborns and it's approved for imaging? Of course, it's not as good imaging as MRI or CT, so it's not used for that anymore, but it was. And so I went to see if anybody had seen effects on the brain, and this guy up at Arizona State named Jamie Tyler had been studying animals. He did some amazing work in animals implanting electrodes and then hitting with ultrasound and seeing electrophysiological effects, or like stimulating here and seeing the paw over here move, that sort of thing. Um, and so I said, well, that's pretty cool. And I I used to run our chronic pain clinic in anesthesia, and so I said to my friends, I said, we should try this on our pain patients because they're depressed. They're all depressed, and this might, give them a mood lift. And they go, yeah, that's a great idea, Hammer. Hammer was my name in anesthesia, as if I put him to sleep with a hammer. And great idea, Hammer. You got a nice shaved head. This was your idea. Why don't you go ahead and do it, and we'll just watch. And they called my bluff, and I said, okay, I guess I got to do it. So one day, at the end of the day, we sat around a table and wheeled in the machine and put some of this goop on my head and held it to my head and turned on the machine. And I didn't feel anything. And I held it there for about 15, maybe 30 seconds. I put it down. I was kind of disappointed. But then about a minute later, I got a buzz, and I was buzzed for like 2 hours. I felt really energized, invigorated, creative. I came back the next day and said, we gotta do the study. And so we did, and it was published in early 2013 in Brain Stimulation, the first study ever on ultrasound effects on human mental states, mental states of any kind, consciousness. And Jamie Tyler, who had done all this work that I had, in animals, I became friends with him later, and he sent me a nice email and said, "Congratulations, you have the first human study." And now there's been hundreds, if not thousands, actually. But the neuroscientists got into it and they took advantage of the fact ultrasound can be focused. And when I did mine, it was kind of scanning, like a scanning ultrasound, and I didn't know what to focus on. Anyway, frontal cortex maybe, but um, but then they said neuroscientists liked the idea you could target the anterior cingulate or the prefrontal cortex or the Q anywhere, but the patient needs an MRI. You need a stereotactic frame to aim at something, and it gets real expensive and technological, but opens the up for intellectual property, and you can invent this and that. So they all got into it, and so most of the stuff, and then they also do it at different intensities. You can do it at high intensity to cause lesions. And I read recently that Bezos and Peter Thiel are starting a big effort to do surgery, particularly neurosurgery with ultrasound. So instead of cutting the patient open, you zap the tumor, and if you use two beams where they intersect in the brain, you could target the tumor and really fry it without damaging anything on the way.
Yeah.
And I think that'd be great, but I don't want to use it for destructive lesions. At
Yeah.
medium doses, you can open the blood-brain barrier, and you can get drugs into the brain that otherwise couldn't get in. And sometimes you put them in these micro bubbles and they get into the brain. You can ask.
Yeah, I just we were got a little bit of time left, and I wanted to get into the implications of what I mean. Basically, if I understand what your theory is saying, is that consciousness preceded possibly life and potentially guided the evolution of human life, life on planet Earth, maybe elsewhere, and that we may be living in a conscious universe. And I was hoping that maybe you could expound upon that idea because it's something I'm sort of fond of thinking about, and it's a tradition that's been alive throughout a lot of, human endeavor throughout human history. So, can you talk a little bit
Right.
about that?
Okay. Well, I skipped over something very important. I was talking about Roger Penrose, because um, when he was trying in the second half of Emperor's New Mind, he was trying to explain the quantum mechanism that, basically the measurement problem, where things can be in superposition in multiple possible states or locations. But when you try to measure them or observe them, they collapse to one or the other. So they say the wave function collapses. And that's using quantum computing. You have multiple possibilities called quantum bits or qubits, and they collapse, all the possibilities collapse to one of the to one value. And that's the solution. So they're using quantum computing. But when you make the measurement, you introduce randomness. First thing Roger did was first say, well, how the heck can things be in multiple states or places at the same time? Superposition and entanglement are the two biggest mysteries, I think, and they're related. And he, for superposition, he went to Einstein's general relativity, which says that mass is equivalent to curvature in space-time. And Einstein did this for big objects like the sun. And so he said, well, light from stars on the other side of the sun from us would be curved by the sun's mass and would be visible to us here on Earth, during an eclipse or something. Even though we knew they were behind them, we should see them. That was his prediction. So in 1919, Arthur Eddington did the experiment, went to a mountaintop during an eclipse, and sure enough, saw these stars that were known to be at that time behind the sun. And the sun's space was curving space-time so you could see them on Earth.
Yeah.
So Roger applied that idea to quantum particles. So instead of the sun, let's go to an electron or a proton or something, and it's going to have a tiny curvature. And then you could imagine that if the proton is here and also here, you'd have two tiny curvatures. So you'd have a separation in space-time geometry. And then you can imagine that if each branch of that kept going, it would have its own universe, and that gives you the many worlds hypothesis. But Roger said, no, the superpositions are unstable, and after time t will collapse to one or the other. And this was the real kicker, when that happened, it would give you a moment of consciousness, now, or bing, when the collapse occurred, after time t equals h bar over e sub g. Basically, the uncertainty principle, and Heisenberg uncertainty principle, indeterminacy principle, and variation of that. And that would give you a moment of consciousness. The reason was the rationale for saying that's where consciousness was that, well, it had to come from somewhere. But the main point was Gödel's theorem that you needed something non-computable that had to be collapsed to the wave function. So if that was your source of non-computability and is essential for consciousness, it also must be the source of qualia and feelings and this and that as a property of fundamental space-time geometry. And that's what I got out of it. In fact, we wrote a paper in our second paper, 96. We specifically made the connection to the hard problem uh, being qualia embedded in space-time geometry. Now, to your question, I think where you were going was let me skip ahead because in around 2000, um, uh, people started discovering these near-death and out-of-body experiences after people had cardiac arrests. And Pim van Lommel in Netherlands and Peter Fennick in UK both did big studies on cardiac arrests and interviewed the patients afterwards, the ones who survived. And 17% in both groups reported these bizarre near-death and out-of-body experiences, which were bizarre, but they were very consistent among different people and between the two groups. And now many groups, probably hundreds, thousands of studies all over the world. And this actually goes back ancient times describing the same thing. It turns out I didn't know that at the time. But what
Plato.
they describe is, a white light, a tunnel, seeing deceased relatives, a sense of calm. And a lot of doctors say, well, it's all hypoxic. The brain's hypoxic. And I've seen a lot of hypoxic patients, and they're not calm. They're anxious as hell and confused. So that doesn't make sense. And we've also seen end of life brain activity when we have these brain monitors on people when they die, that there can be a zero brain activity, then a burst of activity after the heart stopped. So in 2003, BBC did a show about these near-death and out-of-body experiences, and they asked Pim and Peter, well, how would you explain this? And they both said, well, we have no idea, but ask Penrose and Hameroff because they have this crazy idea about space-time geometry. And Roger said, no, thank you, because he's wise enough to not get into these things and he can maintain his credibility. But anyway, I said, well, it could be that when the blood stops flowing, the oxygen runs out, that the quantum information around in the microtubule, kind of dissipates to the universe at large, but remains entangled as something like a quantum soul and can exist in space-time geometry, maybe find another set of microtubules for reincarnation, or maybe persist indefinitely, or the patient is resuscitated, go back in. So
Matrix for the
but
soul, basically, like a medium for the soul to exist within. I
yeah, the
appreciate that.
quantum
Yeah.
soul. That was the name of our paper. I wrote a paper about it with Deepak Chopra, actually.
Wow.
I came up with the science idea, and he had all this knowledge about near-death and out-of-body experiences and mystical experiences and astral projection and all this kind of stuff. All of which, and remote viewing, the CIA did. All of that, as bizarre as it is possible if consciousness is happening in space-time geometry, which is non-local and can be entangled. So I think that's probably what happens actually. And Like a lot of people when I was young, my dream was, well, I want to understand consciousness so I can be immortal and park my consciousness in some kind of device. And I don't care about that anymore. I'll just take my chances the old-fashioned way because I think it's quite likely, and so I don't have to worry about it.
Yeah. One of the things I saw, just sorry, David, do you have a question you wanted to ask? I jumped on
We
you.
can come back to it. I had a question about neurons and brains, but we can come back to that later if there's time.
Okay. I just wanted to ask, one of the things that I'd seen in a previous interview is that you said that in the early periods of the universe, or prior to the development of life on Earth, at least, that there was proto-conscious events that happened,
Right,
and that perhaps an experience of happiness in these proto-conscious events may have led to sort of a snowball effect that eventually manifested in a more complex form of
right.
consciousness. And there's been a line throughout mystical history with philosophers and whatnot talking about the central power of love in the universe. And it seems to me that love slash happiness, some sort of deep motivational feeling that may have emerged in the early universe through these proto-consciousness events may be precisely what those folks are talking about. But
Yeah.
I
I
mean,
wrote a paper about this in 2017. I was asked to write a chapter in a book, A Man and His Nature, about humanity and more of a philosophical approach. And I realized that Roger's objective reduction can happen in space-time without any matter at all, or around a proton or something simple. And it would be a very, uh, and it's probably happening in the microenvironment all around us all the time. In the table, in the air, in the computer, and part of your body that isn't conscious, everywhere. Uh, but because a quantum superposition is going to happen, it's quickly going to run into another quantum superposition. And before too long, it's going to reach the threshold, time t equals h bar over e sub g. It's going to have a moment of objective reduction collapse and a moment of subjective phenomenal experience. However, it would be random because it's in the random microenvironment. It would be disconnected. It would happen, and then that would be the end of it. There would be no memory. There'd be no context. There'd be probably no meaning. So we call it proto conscious, or that was Roger's term, proto consciousness. And so there's uh, this background proto conscious noise everywhere, presumably. Of course, we don't know it, we don't recognize it. It doesn't affect us in any way, but uh, just like all the other quantum stuff that's happening. Um, and it's kind of like metaphorically, like if you go to the symphony and you hear the musicians tuning their instruments before the performance. And it's kind of, to me, who's not musically trained, it's like noise. You hear all these different tone noise, basically sounds, and then the orchestra starts and it's music. So what the brain does, what the microtubules do, is convert the protoconscious noise to consciousness. Now, in this so I wrote this chapter about the origin of life because that's another mystery. And I never really understood how life would start and develop for 100 million years before genes to kind of guide behavior in some way. And even with genes, doesn't, you have problems. But so I wondered whether feelings were there from the beginning. Now, most people would say that feelings emerged much later due to complexity, like with brains. So that means we had to get from for origin of life, I don't know how many billion, I forgot how many billion years, to brains, which are fairly recent. And if you look at all these simple organisms, they're doing purposeful behaviors, intelligent behaviors with their microtubules. But why? What's their motivation? Why would a molecule organize itself or do anything purposeful, fighting entropy um, to survive? Why would it bother to survive if it didn't have any feelings? So it seemed to me that feelings must be there right from the get-go. And with Roger's mechanism, they sure could because he had a mechanism for it, and it could happen between two benzene rings, minimally. You want more and what happened faster, excuse me. But so anyway, I wrote this. I wrote this article, the quantum origin of life, how the brain evolved to feel good. And in it, I described how these OR protoconscious events could be happening in the random environment, and described the primordial soup, operin and haldane, and these aromatic molecules, the same benzene-like, actually dopamine-like molecules. If you put a tail on it, you got dopamine, the pleasure molecule, would be in these my cells, and they would coalesce just like in protein folding and start oscillating, and would eventually have a reach the threshold, have a conscious moment, which at least initially would be proto-conscious, and could be any kind of feeling, but at least occasionally some would feel good. Now you said love and some other things, and I just used pleasure, and I would as kind of a generic term that certainly love would work, and but anything positive that you want more of, right? That's the key.
Yeah.
And that the molecules would then uh, organize themselves to optimize pleasure or optimize love, whatever you want to call it, and would develop and grow, and then they learn to defend themselves and find food to keep going so they could have more pleasure and love. And they'd avoid predators because that feels the opposite of good. And that's how life started to optimize feelings, pleasure, love, whatever you want to call it, and avoid displeasure. And I think that drove early behavior, early life development, and continues. I mean, in a way, everything we do is based on optimizing our feelings over some time scale. Moment to moment, hedonistic or altruistic or long term. Delay your gratification, go to school so you can have more love and pleasure later, however you want to define it. But really, everything we do is to optimize feelings, I think. I think that's probably still true. So,
Yeah.
and it's all embedded and coded in space-time geometry.
Yeah. Did you have any follow-up questions before we wrap it up, David?
It might to see if we could tie all of that together. The question that I was thinking about earlier is just something to clarify. And I think Stuart, you've spoken to it already, but I was wondering if we could close the loop. It always struck me that the microtubule story is it's subneuronal. But when we're talking about consciousness, we're it seems like we're talking about a brain level event. And so you were referring to entanglement and maybe resonance between frequencies. Did you have a mechanism for as to how you go from something that is occurring at the level of the neuron and then coordinating over the entire brain?
Well, entanglement. Microtubules can be entangled, I think, within a neuron and between neurons and probably across the brain, and maybe between a brain and another brain somewhere else in parapsychology, things like that, or between a brain and something. So, entanglement distance doesn't seem to matter. And once you're entangled, you're entangled. So, and Anirban has shown in cell culture that measuring megahertz and gigahertz inside neurons, that there's communication from neuron to neuron in megahertz and gigahertz that controls neuronal firing more so than do the membrane potentials. So it's a deeper system, includes quantum, and somewhere in that kilohertz, megahertz, gigahertz, terahertz, it's kind of like music with different scales, and it can go up and down, and somewhere you're going to cross that threshold and have a conscious moment. So consciousness can be like notes and chords in this music-like hierarchy, and if you go faster, faster, eventually it's more you get more intense experience and more quantum non-local. And I think meditation, psychedelics, all these things take you to a deeper level. You get more non-locality, more quantum, and more connected to stuff outside you, in your brain and outside your brain. And eventually, when our bodies die, we probably just go there completely and are distributed. And I think there's a good chance that consciousness in the brain is a hologram, and what you need is interference patterns. So one thing that is uh, so when we said, okay, 10 megahertz is the frequency for ORCH-OR events, but that's too fast for cognition. I mean, most of what we do interacting with the outside world is more in EEG frequencies, 10 hertz, something like that. So how do you get from megahertz to uh, 10 hertz, let's say? And I remember having this discussion on the phone with Roger, and we were writing our 2014 paper. And by then, Anirban had discovered the megahertz oscillations, so we knew we had evidence for that. We had to figure out how we get from megahertz, and also having the very fast megahertz made it easy for us because you only had to avoid decoherence for 10 to the minus 7 seconds, as opposed to 25 milliseconds if you want to say it was for 40 hertz, which would have been unrealistic. So I remember he said. He said, well, maybe there's negative resonance. I'm going, negative res He says, like interference beats, like in music. And I said, oh, so you mean like if there's two microtubules, and strangely enough, in dendrites and soma of neurons, and only in dendrites and soma of neurons, in all the biology, the microtubules are not continuous and in the same polarity. They're in mixed polarity. So one's going up, one's going down. So if this, and they're in the same voltage, this one's going to oscillate slightly different frequency than this one, and you're going to have a beat frequency. So this one's going to be at, let's say, 10.004 megahertz. This is going to be 10.000 megahertz. They're going to have a beat frequency at 40 hertz, and that's going to give you. So we proposed that the EEG is actually due to beat frequencies of much faster vibrations than microtubules, and people thought we were totally out of our minds. But then I would answer, well, how do you explain EEG? Because after all these years, we really don't have an effing clue what EEG means and what it's all about. There's an excellent paper by a guy named Michael X. Cohn from 2019, something like the EEG, where does it come from? What does it really mean? And it points out all the stuff that we don't really know. I mean, yeah, I read Busaki's book, The Rhythms of the Brain, and alpha comes from this pacemaker, and theta comes from this circuit, and this and that. He had no idea about 40 hertz, but they don't really know. And there's no grand unified theory of EEG except ours. We would say that EEG is actually B frequencies of much faster vibrations than microtubules. Now, nobody even acknowledges that, but they don't have a better explanation for what EEG is, unless it's just all random and doesn't don't relate to each other. But that would seem a little unusual to me, that it's not some kind of organized system. They're just looking at the wrong frequency. I think you need to look faster.
Love it. Let's end it with one more brief question. What are you expecting coming up in terms of your theory that may provide more progress, or what's the thing that you're anticipating most in terms of proving? Is there any chance on the horizon that that might be coming your way, or how do you see that working out?
Well, we keep working and looking for more evidence. Anirban is building there's this big thing now with AI. Can AI be conscious? And they're trying to convince us that it will be. That even Chalmers is throwing the heart problem under the bus and says that it's inevitable. Of course, he confuses carbon chemistry with organic chemistry. But anyway, um, I don't think silicon can do it. But if you really, I mean, the best way to do it would be to copy the brain, right? So what Anirban's doing, he developed something called brain jelly, which is a organic gel that self-organizes into helical oscillators. And in his view, the key to it's kind of like microtubules, where you have helical, and you mentioned DNA, helical oscillators, and then the oscillators, the aromatic rings line up and they oscillate, and that you get Froelich coherence and stuff like that. So this brain jelly self-organizes into a system of helical oscillators that interact on their own, and he interfaces with them with I think microwave and optics. I forget which is the input, which is the output. And he's building and he's got a 1024 qubit design for an organic, warm temperature, self-organizing quantum computer, organic quantum computer that he's building. And he just got another 25 million from the Indian government. And that's not a lot of money in this AI, quantum AI thing. But India, they look at it as a way to kind of leapfrog over all the expensive cold temperature Google AI type approach, where you have to ultra cool everything, and then you lose any connection to biology. So they think they're going to leapfrog that with their organic warm temperature. You don't have to cool it. Solves a lot of problems. And not to mention the fact that these AI systems to power these LLMs use an enormous amount of energy, and we've already got enough problems with energy in our world. So I like the organic approach, and predict that that will be the first potentially conscious AI. I'm sure there'll be a period of they're claiming it's conscious and we don't know, blah, blah. But I think at least whatever the output of that thing will be, it'll probably be sensitive to anesthesia because the quantum oscillations of the aromatic rings are essentially what happens in our brains. And maybe you asked me at the beginning, but people always ask, well, don't let's have a definition before we explain consciousness. And there's no good definition. I mean, the best definition I have is what goes away with anesthesia and comes back when the anesthesia is gone. So if Honor quantum computer does that, then I guess we have to decide whether, is it conscious or not? And I don't know. We'll have to fight about that at that time. But that's going on. I think the other thing so I'm working now in astrobiology. When I wrote that paper about the origin of life, it was picked up by Dante Loretta, who's the planetary scientist here at the University of Arizona, and he was in charge of this mission to an asteroid called Bennu. It was a 20-year NASA mission, brought back organic molecules
Yeah.
that were analyzing for quantum oscillations. We see quantum oscillations in these, and they're basically the same aromatic rings that we've been talking about. The space is full of them. They're produced by stars. Interstellar dust is full of aromatic rings. They're floating around. You can wipe them off the windshield of the space station, and they're probably floating down on us now. And there's almost no doubt that they are the origin of life. Aromatic rings from elsewhere, because initially they had to come from space, panspermia and stuff like that. But they don't have to be organic. You just need the aromatic rings. So anyway, while we're waiting for the Bennu samples, there's a molecule that came from a meteorite that fell in Australia in 1969 called the Murchison molecule, which is kind of a bouquet of aromatic rings with 35 rings. And it was kind of a two-dimensional because of like a pressed flower. This picture had been in the literature since 1969. And we sent it to Anirban and said, could you model this in 3D, kind of like protein folding? And he did, and it turned into an oscillator in petahertz. And we think it might be the ground state of a time crystal as the origin of life. So if you started with an oscillator and it started having protoconscious moments, including pleasure or love, then it would grow and get larger, but then it would also get slower because you have more mass. So you not only have petahertz, but then you'd have terahertz, and then it get bigger and you'd have gigahertz, and then eventually you'd have a microtubule. And so we're Anirban and Dante and we just are submitting a paper on microtubules as time crystals and what that would solve for consciousness and life. And I think that's going to be so. Time crystals well, spatial crystals, they repeat in space. Time
Right.
crystals, you have dynamic patterns that repeat in time, so you can have the same dynamics in, let's say, hertz, kilohertz, megahertz, gigahertz, terahertz, which is exactly what we found, or what Anirban found in microtubules. And time crystals were proposed theoretically by Wilczek, who won the Nobel Prize for something else, and have been implemented in the last few years in various types of cold quantum systems. And we're proposing it in a warm biological system, which is a big ask. But we've already got the evidence. We've already got the data. I mean, that came first in this case, and we had to figure out, well, what are we dealing with? And we figured, well, shit, it's a time crystal. That's pretty much the definition. So we hope and think that the time crystal concept will turn out to be very important in the origin of life and consciousness. And maybe the final point I'll close on is that I was the relationship between life and consciousness, and somebody maybe it was Deepak or somebody at Deepak's conference told me in the Upanishads that um, consciousness was there first and got bored and invented life as a vehicle for more
Mhm.
experiences. And I think
Yeah.
that might sort of be true, that life is a vehicle for consciousness. And it's not that consciousness evolved from life. Life evolved to optimize consciousness.
Yeah.
I'll leave it at that.
And it turns the whole question on its head, right? I mean, it's like
Yeah.
you don't have to worry about all the complexity that gives rise to consciousness because consciousness was.
Complexity is just an overused, abused term. I mean, it's not a solution. It's not the answer to anything. And I spent a lot of time in complexity chaos theory with a guy named Alwyn Scott, who wrote a wonderful book called The Stairway to the Mind about emergence theory and consciousness. And when I started working with Roger, he had been looking for the Davidoff soliton his whole career, and he couldn't quite buy the fact he never found it, which is a quantum mechanism. So he was kind of resistant to that. So we parted ways scientifically. We remained really good friends. But so I'm familiar with emergence and complexity, and I think they're just beating it to death to explain something that they cannot explain any other way.
Well, I appreciate your time, sir, and thank you for a fascinating discussion and a whirlwind tour through all of your work. So thank you so much.
Thank you both very much. My pleasure. Good job. Good questions.